Hi everybody, welcome to Termination of Replication. Here I am. Okay. Hello. Okay, so let's start with termination of replication in E. coli. We're not going to get into this in any elaborate detail, but we will go through this, so you get the idea. So we have our circular E. coli chromosome shown here in blue. We have the origin at the top here at 12 o'clock. We have bidirectional replication forks extending away from the origin. And what the chromosome includes on it are these termination sites that are located roughly around 3 o'clock on one side of the chromosome, 9 o'clock on the other side of the chromosome, and then there's another big cluster of them down here close to 6 o'clock. Okay, so what happens here? So termination sites have these associated TUS proteins, or termination utilization substances, and they act to slow down and help with the timing of the moving replication forks, because we have these two separately moving replication forks, and we do want them to get to the bottom at 6 o'clock around the same time, so we get everything copied around in sync with each other. So we have our replication fork 1 is passing through down on this the 1 to 6 side of the clock, and it hits the first cluster and it slows down a little, and then it keeps getting, and it finally gets through the second cluster of termination sites and is slowed down and stopped further. And the same thing happens on the 12 to the reverse side of the clock. So we go down here, we slow down a little, we slow down some more, and what's interesting about these TUS-TER units is that they are polar. They only work in one direction. So let's just stay here, that we both of our replication forks get here, they stop, and we have completed replication. And what they do, what the TUS termination centers do, is prevent the opposite replication fork from proceeding. So even though they slow down in one direction, the other one they just obliterate it. So I think a good image to think of them as kind of like these one-way speed bumps that you can see in parking lots. So you can imagine one replication fork, the one that's permitted to go through them at 3 o'clock and 6 o'clock, the replication fork that can go through it, but it gets slowed down a little. But a replication fork coming from the opposite direction can't pass through it at all. So there's a little bit of redundancy in the system, as many biological systems are prone to have, but so the replication fork on the other direction is completely obliterated. And when we get to the end, we've gotten 2 copies. The replication forks have met in the middle down here. It's completely blocked now. And all that's left to do is we have 2 complete chromosomes, but they're kind of tangled up a little bit, which you can easily, I hope, imagine, because of we have 2 circles and they've been kind of wrapped around each other as they've been replicating. So our topoisomerases, or as they're called in E. coli, gyrases, are used to help disentangle the overlapped circles. And once each daughter chromosome is disentangled, they can go on to their next daughter cell and repeat the process when the time comes. So that is termination of replication in E. coli. So now we're going to move ahead and talk about the termination of replication in eukaryotes, in any organism that has a linear chromosome. So to start, I want to bring us back to week 1 for just a second, where we talked about all the components in eukaryotic chromosomes. And this particular pie chart represents all the components in the human genome. Remember that? And this small little segment here, that's actually the coding elements, and everything else is non-coding. That means it's doing something else functional. Many of these functions have been elucidated, and but it's not coding. And you can see indicated here in green that these long repetitive elements include the categories of centromeres and telomeres, comprise a pretty substantial portion of the genome. Now, the reason they're called long repetitive elements is because they're long, the sequence in the telomeres, at least, is actually simple, but it's repeated many, 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 many times. And we'll look at that in just a second. So why do we need special sequences at the ends of chromosomes? Because there are a couple problems that are associated with having a linear molecule that's being replicated in a cell. The first of these is called the end loss problem, and we'll talk about that in just a second. And the other one we'll talk about later is the fact that if you just had plain double-stranded DNA as if it were made by DNA polymerase with nothing, or Taq polymerase with nothing to distinguish the end of it, it would be subject to DNA repair machinery, and we'll talk about how that's resolved also. But first, let's talk about the resolution of the end loss problem. So what do we mean when we say the end loss problem? The end loss problem is a problem that is associated specifically with lagging strands. So let's start there. So we can see here the model of DNA replication. So we have the parent molecules up here. We have a replication bubble with two replication forks going in their respective directions. And we can see that the leading strands make it to the end without a problem. They just zip right along. They've been primed once and they go like that. The lagging strands are busy chugging along with their Okazaki fragments being made every kilobase or so. And so you can see the RNA primer from the Okazaki fragment, and then the DNA, and then RNA primer, then DNA. And as we learned that when Okazaki fragments are resolved, let me skip ahead to the next slide for that. That what happens when the Okazaki fragments are resolved is that that piece of RNA gets degraded. So RNA comes in there, degrades the RNA primer from each Okazaki fragment, then DNA polymerase 1 or alpha fills in with dNTPs, so you get completed DNA molecule. So now we can see the Okazaki fragment's been processed, and we had now we have a complete lagging strand. This half was the lagging strand on this one, except that there was nothing to prime the material at the end here. So we've lost about a kilobase material at the end of each lagging strand. And we've also lost down here. So we go on to the next generation. There's no template left. And then we're going to lose another kilobase because the same problem will happen again and again and again. So we can't afford to be losing all a kilobase or 2 kilobases per chromosome per replication cycle. That would be pretty disastrous. So we need a solution. Okay, so now let's take a little bit closer look at a telomere. So here we have a chromosome shown in the upper left of the slide. Nice little metaphase chromosome. And you can see the green ends indicate where the telomeres are. Okay. Telomeres, here's a sample of some telomere sequence. You can see how repetitive it is. So this is considered 1 unit of telomere repeat. In humans, TTA-GGG, TTA-GGG. And that repeats it over and over and over and over and over and over and over again. The proteins that interact with telomeres are TERT, and then there's some proteins that are associated with that called telomerase-associated proteins. TERT is a reverse transcriptase, so that's telomerase reverse transcriptase. And telomerase also has a special little friend in it that is a little piece of RNA. And this RNA actually acts as a template. So functional telomerase is composed of TERT, reverse transcriptase, and it's built-in RNA template. The telomerase-associated proteins are involved in regulating the length of telomeres and forming a structure we'll talk about in a few minutes. Okay, so let's see what telomerase is doing at the end of the telomere. So on the top here, we have an incompletely copied chromosome. You can see the lagging strand, the parent strand is complete on the top, or I guess it's a parent strand, not a lagging strand, and then the lagging strand is incomplete on the bottom. And it can't be complete because there's nothing to prime it. Telomerase comes in and it actually binds to the final 2 nucleotides of the parent strand. This shows it binding a few more. And then it extends it acts as a template, and so then we can increase the length of the parent strand, and that goes on, and that repeats. So the telomerase binds, extends the length of the parent strand, dislocates, moves down, and it makes it longer and longer, and that repeats several times. Eventually, there's enough additional parent strand, another primase lands down here, independent of a complete replication fork, and allows a new primer region to be synthesized, RNA primer to be synthesized, which is down in here. So this would be RNA that was primed by primase. And then DNA Pol alpha can synthesize and complete the rest of the molecule. And of course, we'd still need a little ligase to come in and glue that little bit there together. So in that way, we've solved the end problem. So what's happening is we have this repetitive end where actually it's kind of being degraded and extended in a kind of not perfect equilibrium. So the telomerase is trying to perpetually extend the end of the telomeres, and we're still losing some at the end, but we've added additional stuff to create a buffer between the actual coding sequence and the telomere sequence. Okay, so we've been talking about the two problems that happen at the end of linear molecules. One of them is the end loss problem, and I think used to solve that. But now let's talk a little bit more specifically about this other problem here. So that is the fact that if there was nothing done to distinguish the ends of chromosomes, they would just look like double-stranded breaks. So next week we're going to be talking about, or next coming lectures, since we're kind of self-pacing, are going to be talking about different types of DNA repair. And there's many versions, there's many specific types of DNA repair, as there are many different types of DNA lesions. And a very simple type of DNA repair, lesion is when both strands of the phosphate backbone break. DNA repair. And so our cells have the ability to simply recognize two pieces of DNA, if there's a double-stranded break and they're right next to each other, like this, and glue them together. And that process is called non-homologous end joining. And it's represented by the initials NH, BJ. It's obviously a ligase-mediated event. And so if we got to the ends of our chromosomes during replication and did nothing to make them look anything then like the ends of DNA, they would float around in the cells and they look like two ends of DNA with a double-stranded break. Our non-homologous end joining machinery would say, oh, we got to glue those guys together. But what if this end came from, let's say, chromosome 2, and this end came from chromosome 11? Or, I guess even worse, what if this was one end of chromosome 2 and the other end of chromosome 2, and they loop back together, and suddenly we had a circular chromosome? So we have to do something to make sure that we've distinguished the ends of our chromosomes to be unique. So we've solved the problem of losing material, but now we got to just also make them look unique so we aren't randomly gluing chromosome pieces together. So that is the next piece of what's distinctive about telomeres. So the telomere ends are not in the form of nucleosomes. They have other proteins that bind and protect them and package them. And these proteins are shown in this figure here. There's these various TERF proteins. There's TIN2, TPP1, RAP1, and POT1. You can see that TERF1 and TERF2 bind directly to the repeat sequences within the telomere sequence, and then POT actually binds to single-stranded versions of the telomere sequence up here. And you can see that they're here in all these proteins together are called shelterin, and you can see how they kind of work together to create this unique structure at the end of the telomere. So there's multiple copies of it going along at the end of the telomere. But POT1 actually has a kind of unique function in this, in that it helps regulate the length of the end of the telomere. POT1 is a weak inhibitor of telomerase. So every time you have one, if you have one unit of POT1 on a telomere, it's going to slow down telomerase a little bit. As you add more and more units of POT1, you're going to increasingly, let's see, I'm circling the right one as I go along here. I think you're getting the idea, though. You'll increasingly slow down telomerase more and more. And at some point, the additional POT1 proteins have added enough, enough POT1 proteins have been added to the end of the telomere to signal to the telomerase that its job is done, and it's made a long enough end to the end of the telomere. The next thing that happens, if we're not going to get into too deep into the molecular details, but you can see it must be pretty cool, is the end of the telomere actually loops back around on itself here. So you can see that I'll draw on it up here, so you can see this at the end of the telomere. Once we've POT1 tells it that it's gone far enough, you can see it kind of looping back on itself. So let me erase it, maybe just shorter. I just think that's so neat. There. Red, so you can see it looping back around on itself like this. It's almost making a little knot at the end of the chromosome. This structure is also referred to as a T-loop, as we'll see in the next slide. So you can see here, this is actually EM now. Yeah, there we go. And you can see that this is not an entire chromosome at the end. They're much longer than that. This, you can see from the scale bar here, it's about, a kilobase is about whatever that unit is in this picture, a centimeter, half a centimeter. So this thing may represent about 10 kilobases or so at the end of a chromosome. And you can see the T-loop here. So that is a covalently, or combination of covalently and hydrogen bondedly created protein DNA structure. So now we have a distinct structure at the end of each chromosome. So instead of going out here and hanging a simple broken end, it's looped back around on itself, and we've kind of, and now this chromosome end can no longer be accidentally recognized as broken DNA. It's got a unique structure, and that helps protect and maintain the end of the chromosome and stabilize the whole genome, so that we don't have chromosomes randomly getting glued together when we don't want them to. Okay, so I'd just like to draw your attention to the fact that there are, I believe, 2 Nobel Prizes awarded for telomeres. The most recent one was in 2009, where they really cracked this problem with the end problem and characterized how telomerase works and showed a lot of things about its sequence specificity. They transferred different telomerases from different organisms to show how the different pieces of it work. It's very interesting stuff. And it's always nice to highlight women scientists. And if you want to, I have later on a link for, you can watch Elizabeth Blackburn present her story of how she figured out that with her colleagues and that worked with her the telomere problem. I just want to review a couple telomere facts to make sure I covered these things. One is I didn't talk a lot about the sequence. They're generally, all telomeres are generally GT rich. I pointed out in that one slide, the human telomere repetitive sequence, which repeats over and over again, is TTAGGG. You can see another example for meese is TTGGGG, so they're not the same, and that's the case for different organisms. Telomeres are long, but they vary in length in both organisms and individuals and throughout our lifetimes. So when we're younger, our telomeres are longer. As we get older, our telomeres get shorter. Some people believe that there's a correlation that if you can make telomeres longer, you will live longer. I'm not 100% sure about that. Telomere length is affected by diet, exercise, stress, and disease. That has been shown. So as I said, telomeres get shorter as we age. In contrast, though, if you look in many cancer cells, their telomerase is overactive. In most cancer cells, If you look at them division after division, even though in normal cells our telomeres get progressively shorter, so if you would draw, like here's the end of the coding region of some chromosomes. As we age, our telomeres get shorter. So, like, this is the length of telomeres as the cell is repeatedly dividing. And eventually, once the telomere is short enough, the cell will stop dividing. So you can imagine some type of feedback with cell cycle, and that would tell the cell to go into G0 and be in senescence, right? However, in cancer cells, if you look at telomeres after many divisions, they're pretty much the same length. They just stay the same length. These are the telomeres in cancer cells after many divisions. So, for instance, if you were to look at HeLa cells, which we've mentioned many times, HeLa cells now and the HeLa cells in 1930s have this pretty much the same length telomeres. As I mentioned, there are studies that indicate that telomere length is affected by diet, exercise, stress, and disease. And there are companies that sell supplements that are supposed to help your telomere length, but that's probably just a bunch of bunk. Because you can imagine that in our cells there's a lot more things going on than just the length of telomeres in determining the health of the cell. And the last thing I want to draw your attention to is the Nobel lecture by Elizabeth Blackburn. I thought you might enjoy watching that if you have extra time. So, just to quickly sum it all up, in terms of replication termination, E. coli has TER sequences and that are bound up with TUS proteins. They act to prevent replication forks from passing through each other. And the replication is completed around 6 o'clock on the bottom of the chromosome, 180 degrees opposite of Ori. And once both halves of the strand are replicated, the two completed daughter strands are resolved by some gyrases. Linear chromosomes have two problems. One is loss of information on the lagging strand, and then also that if we don't do anything to have a unique structure at the end of our linear molecules, our DNA repair machinery might randomly repair those ends and kind of mangle up our genome. So telomeres solve both of these problems, one by self-priming long stretches of repetitive DNA through the use of telomerase and its built-in RNA primer, and then also our telomeres form into a unique structure so that they are clearly indicated as ends and stabilize our genome. And that's that.